Today I'm just gonna cover some key points for tomorrow's biology CA. Tomorrow we have the immune system, the excretory system, some grid questions from the circulatory system, and we also have photosynthesis from last year's book. So I do have some questions for that. I'll show you guys that in a bit after we finish this. So let's start. We have the immune system, starting with pathogens. A pathogen is a microorganism or a virus that can cause diseases in living organisms. When a pathogen enters in the living organism's body and invades it, it is considered an infection. And when the body is able to resist and fight back this infection, this is called immunity, which is carried out by the immune system. Now, the organism that has been invaded by the pathogen is called the host. They may ask you this, they may not, but just in case, just know the person being infected by the pathogen is just called the host. Now, diseases caused by pathogens are transmissible, or you can say infectious, while diseases not caused by pathogens are non-transmissible or non-infectious. For example, flus in general are infectious and transmissible, meaning you can pass them on to your friends or family, while diseases like cancer or diabetes are non-transmissible because you cannot pass these on to people. They are you cannot pass it on if you come in cross-contamination or anything. Now these can be directly or indirectly transmitted. So as I just said, the transmissible diseases can be passed on from the infectious person to the non-infectious person. This can also be caused by contaminated food, drinks, or interactions with vector organisms. Now, when we talk about contaminated objects, it's just if the sick person touches object, it's advised not to go near what they have contaminated, because this can cause the other person who isn't infected by the pathogen already to be infected. Now, if you follow your personal hygiene well, this can limit the cause of being infected by pathogens. So just brushing your teeth in general, which can limit the pathogens entering from your mouth, which is initial ingestion. So that's one thing. Taking showers regularly, all that, just basic hygiene. Now, when we talk about proper sewage treatment, it's very essential to limit pathogens in water. So, say if sewage isn't treated properly, that can cause an increase in pathogens in water. Landfills are places where trash or waste is just dumped. So that's one big place where several pathogens and microorganisms can grow and possibly create new variants. For this reason, we need proper management required for landfills. Now, the innate immune system, this functions with mechanical and chemical barriers, and this is also known as the first line of defense by the immune system against pathogens. So basically, mechanical functions include your skin, your cilia, and your nose. And chemical barriers can include things like your stomach acid, your mucus, your tears. So yeah, these are some barriers known as the first line of defense by the immune system against pathogens. Now, what happens if pathogens cross these lines of defense? The second line of defense is triggered, and that includes inflammation. So during inflammation, you experience redness, swelling, pain, and possibly heat. Now, white blood cells, when this happens, the white blood cells leave blood capillaries. They move to the damaged tissue to kill the pathogens by phagocytosis. And as we learned earlier, phagocytosis is the process by which phagocytes engulf the pathogen. So an autoimmune disease develops when the immune system fails to recognize present antigens of our body. So, basically, just when your immune system can't recognize your actual antigens of your body and ends up engulfing and killing them. So, these are the diseases the autoimmune system can cause. So we have type 1 diabetes, Hashimoto's thyroiditis, and rheumatoid arthritis. So that's it for the immune system. For the excretory system, excretion is the removal of metabolic waste products, substances in excess, and toxic compounds. Now the kidneys will excrete through the urine. Skin excretes water, salt, and urea in sweat. Your lungs excrete carbon dioxide and water. So bilirubin is a product from the breakdown of hemoglobin. Basically, it's from worn out red blood cells, and this is then normally excreted by the large intestine. Urea is produced in the liver as the result of deamination of excess amino acids, which is then released into blood to be filtered by kidneys, then excreted through the urethra. Now the organs in the excretory system, we have the parts as well. This includes the cortex, medulla, and renal pelvis. We also have the ureters, which are tubes that carry urine produced by kidneys from the renal pelvis to the urinary bladder. Both kidneys are made up of thousands of tiny tubules called nephrons. And nephrons are the functional unit of urine production in kidneys. So I'm just going to repeat that again. Ureters are tubes that carry urine produced by kidneys from the renal pelvis to the urinary bladder. Both kidneys are made up of thousands of tiny tubules called the nephrons, and nephrons are functional units of urine production in kidneys. Now there's two steps of urine formation: filtration and reabsorption. During filtration, the water and undissolved substances like mineral salts, glucose, and urea are filtered out of the blood into the renal capsule within the nephron. Within the nephron. For reabsorption, substances needed by the body are reabsorbed from the nephron tubules back into the bloodstream, which conserves the useful compounds in case your body needs it for later. Now I'm just going to repeat that because it is a bit confusing. The first step is filtration, and during filtration, water and dissolved substances, which include mineral salts, glucose, and urea, are filtered out of the blood into the renal capsule within a nephron. Now for reabsorption, the substances needed by the body are reabsorbed from the nephron tubules back into the bloodstream, which then conserves the useful compounds your body can use later on. Now your body regulates a urine amount to its present needs, so it does possibly keep some in case it needs. Like if it needs it, then it's gonna keep it or store it possibly, then excrete whatever is not needed according to the body, and then it gets excreted. The urinary bladder stores urine temporarily before it's discharged to the urethra, basically just released from the body. Some kidney diseases can cause kidney failure or reduce functions in the kidney. Now, two treatments for this are kidney dialysis or kidney transplants. Now, we already know kidney transplants are when a family or anyone who matches your kidney can give you one. Yes, it is possible to live with one kidney. Dialysis is the process by which urea, excess salts, and water, unwanted substances, are removed from the blood of a patient suffering from the kidney failure. It is performed using a special machine called a dialysis unit or a kidney machine. Kidney rejection can occur when the patient's immune system recognizes antigens present on the surface of the transplanted kidney cells as a foreign, and it destroys the transplanted kidney. When kidney rejection occurs, this can just possibly just ruin the entire point of the kidney transplant, because Your kidney is failing to recognize the antigens present on the surface of the transplanted kidney, and it thinks it's another one, so it destroys the transplanted kidney, which means it causes even more kidney failure as it is. So, in general, we have the kidney's cross-section here. This is the cortex, this is the ureter, this is the medulla. We have the renal artery here, the renal vein here, an arteriole, a venule, capillaries. We have the pyramid here, and we have the renal pelvis. You also need to make sure you memorize this part because they'll ask you about this diagram. We have the aorta here, we have a vena cava here, the renal artery, the renal vein. This is the kidney. These are the ureters. They connect to the urinary bladder and then so on to the urethra. So, moving on from this, we have, I'll just go to the photosynthesis chapter. Now, doing the CQDs for this is much more efficient because they seem like they have questions which may come in the exam tomorrow. One of them includes the chemical equation or the word equation of photosynthesis, which is just carbon dioxide plus water gives you sugar, also known as glucose, plus oxygen. Then we have chlorophyll and light energy here. Just memorize the equations and you're good because they're probably going to ask you that. Most of this is memorizing and it's pretty easy to understand. I have a feeling this question may come. So here in this question we have name 3 factors that might affect the rate of photosynthesis in plants and specify the factor affecting the rate of photosynthesis at X. Now the first one we have here, temperature affects the rate of photosynthesis. Increasing the temperature up to 40 degrees Celsius would increase the rate of photosynthesis. Concentration of carbon dioxide is important to be maintained high for the dark reaction to occur. As we increase light intensity, the rate of photosynthesis will increase till the point where all chloroplasts are occupied or no more carbon dioxide is available. So at this point, the rate will stay constant or it might decrease. And then at point X, carbon dioxide concentration is the limiting factor. So, let's see, there is another question. They may bring this question as well. The CQDs actually have a lot of questions that may come in the AMS tomorrow. This is definitely going to come. Plants need magnesium to make chlorophyll, and they use nitrates to make proteins. Just memorize that. This experiment specifically I wanted to mention. It's for the leaf. This I have a strong feeling actually this may come in tomorrow's AMS. So just memorize the experiment exactly how it is because they bring weird questions and it can be a bit confusing. So memorizing it off the CQDs is better than just going back to the old book and reading it because the CQDs, I believe the question may come from it exactly. Not in this way, in multiple choice. So here we have what is the main storage form of glucose in the plant. It's stored as starch. 2 evidences that photosynthesis has occurred. It's either the release of oxygen as a waste product or the production of starch. What test shows the evidence of photosynthesis in a leaf? Testing a leaf for the presence of starch using diluted iodine solution provides evidence that the leaf is photosynthesizing. The appearance of a dark blue color indicates the presence of starch. Now describe how a plant is destarched. A plant is destarched by leaving it in darkness for 2 or 3 days. In a closed cupboard. This will cause any starch present to be converted into glucose. Glucose is used up by the plant for respiration. So here it's in a table. So they will definitely ask this. in the exam about the leaf and how it's going to be after the sunlight. I believe last year they asked a question related to this in the CA, so they may bring that question exactly. Yeah, I believe it was this. Yes. So memorize that. Oh, and this specific experiment may also come. It says state whether or not photosynthesis and respiration are happening in each of the above test tubes. In test tube A, respiration is happening and photosynthesis is not happening. In test tube B, photosynthesis and respiration are happening. In test tube C, respiration is happening and photosynthesis is not happening. In test tube D, respiration is happening and photosynthesis is not happening. So, just memorize this experiment. It's most likely to come tomorrow if it comes from a prerequisite question. We also have this experiment from last year, which again, it's better to look at them in the CQDs. I doubt this may come, but again, just go over the CQDs from last year for photosynthesis. It has a lot of questions which may come. Now when we move on, I have some questions in the CA quiz. We have number 5. Yeah, this is exactly what I mean. They may bring the question like this. So it's in the CQD. If you memorize it like this, it's best to do that. For the circulatory system, make sure you still remember the vessels and what their functions are, because they will definitely ask us this again in the AMS, considering they did in a lot of questions last week. The previous week when we had it. We have 8. I have a strong feeling they are going to ask a lot about antigens. Now, has A and B antigens but no antibodies? This is blood group A. The blood group, people with blood group A, they have A and B antigens but no antibodies. Antigens are the markers on the cells. They have A antigens and B antibodies. This is blood group A. Blood group A obviously has A antigens, but they have B antibodies. Just make sure to know that in A and B, their antigens are of their blood group, but their antibodies are of the opposite one. So for antibodies A, so antigen A and antigen B, has an antigen A and antibodies. It's antibodies. Has antibodies A and antibodies B, but no antigens. This is blood group O. Blood group O consists of both antibodies A and B, but they have no antigens if you look at the structure of the cell of a blood group O. Has antibodies A and B. Antibodies A and B antigen. Blood group B has antigens B, and their antibodies are A. So the opposite of blood group A, they have antigens A and antibodies B. So next, I have 10 here. This is why you should memorize the cross-section. They are most likely to bring a lot of these cross-section questions in the CA tomorrow. Here we have one, if we go back here, like this one, just memorize them. X is the renal vein, Y is the renal artery, R is the urethra, Z are the ureters. Then I have 11. Again, they're gonna ask about the cross-section. Like I said earlier, memorize the cross-sections of the kidney because they're gonna ask you this. Next, 12. This is the process of deamination. So make sure you memorize this. They're gonna ask you this maybe as a fill in the blank, maybe in another way. So excess amino acids are not stored in the body, they are carried to the liver where they are deaminated by the removal of their amine NH2 group. This results in the production of toxic ammonia, which is converted into urea and released into the blood to be filtered out by the kidneys and excreted in the urine. The remaining acid group energy-containing molecule is converted into glycogen, then into glucose, and used for cellular respiration as a source of energy. Next I have 21, 23. 123. So they may ask you the further functions of the liver. It's producing urea by the deamination of amino acids, storing glucose in the form of glycogen, making cholesterol for cell membranes, storing iron coming from red blood cells. This is the temperature, limiting factors, light. Okay, yeah, so memorize this because it was on the CQD also. It's from last year's chapter. So 34 and 36. 34, 36. Oh yeah, so 34 is the equation of photosynthesis. Again, memorize that. 36 is these kind of questions come about in the AMS. So memorize these as well. It's mainly memorization, and if you understand it, then you may not even have to memorize it, but this kind of question will definitely come in the CA, so make sure you know them. Then, okay, this question specifically, they always ask for the order of processes.